So welcome everyone, and thank you for joining today's TCG Digital webinar on how knowledge graphs use to transform and get siloed data AI ready. I'm Angela Bauch. I'm Director of Product Management. I'm responsible for generating solutions and supporting pre-sales activities using knowledge graphs and ontologies. And yeah, there's also Eli, my colleague, with whom I will share this webinar. I will show one use case and Eli will show the second one. And now I will share my screen and start the webinar. So in life sciences, the challenge isn't just the amount of data, it's the fragmentation. So information is scattered across regulatory, clinical, and research systems, making it difficult to connect the dots. And today, Eli and I will explore how we can structure this fragmented data, connect it, and turn it into usable knowledge. And because AI needs more than just language models, true intelligence often comes from context, traceability, and domain expertise, and not only from text. Ultimately, it all nails down to FAIR, which is the acronym for findable, accessible, interoperable, and reusable. So now let's have a quick look at the main challenges we see in life sciences data and compliance today. First, siloed information. So regulatory, clinical, and research data live in separate systems, causing duplication and inefficiency, which is our next challenge, manual and inefficient processes. So compliance checks are slow, document heavy, and often prone to human error. And the third challenge is regulatory complexity. It's difficult to align data and documentation with industry standards like IDMP, CDISC, or MEDRA. And how do we address these challenges? So here on the right is our approach. So we add a contextual knowledge graph layer, a semantic layer, linking in-house and public data into one FAIR foundation. Or we add AI-powered semantic search and analysis, enabling deeper insights across multiple sources. And third, we have a metadata-driven compliance, ensuring traceability and audit readiness. And all these solutions are powered by the same semantic AI platform, mcube. So once the foundation is in place, many high-value use cases can be realized efficiently. This is shown here in the next slide, where at the bottom we have mcube, our technology platform, the semantic AI platform, and instead of building isolated solutions, we create an ecosystem where data, knowledge, and intelligence, they flow seamlessly. So in the middle, we see the different use cases we're going to address today. So whether the goal is here at the left, regulatory compliance, or in the middle, FAIR data metadata for clinical insights, or at the right, AI-powered master data generation, the same platform components can be used. So I will present transforming regulatory documents into structured knowledge, which has the business value of smarter compliance. And Eli will then take over the FAIR metadata clinical study insight use cases, which allows you to get connected knowledge, go for faster decision and accelerated submissions. And as mentioned, the same platform components can be reused, which is shown here in the next slide, where at the center we have this semantic layer for harmonizing data across different systems, which can be here as shown in the bottom layer, the integration layer. We can integrate data physically, like the data which we're going to extract from regulatory documents, or we can set a semantic layer on top of a LIMS system or any other database you might have in your organization, or we just integrate virtually data from another database without data duplication. On top, we have then the consumption layer, where we can build intuitive web interfaces for reporting and exploring, or for AI analytics. And we can also add AI models that extract meaning and context. And now, before we switch to the live demo, first a quick overview, a schematic overview of the solution on how we extract data from regulatory documents to structured knowledge. Here, as an example, we took SMPCs, which are summary of product characteristics. These are standardized laboratory documents containing key details on drugs indication, dosing, contraindications, formulations, and so on. And these are essential both for regulatory and clinical work. But extracting these structured data from these documents manually is time-consuming and often error-prone. So how do we turn this unstructured content into reliable, structured, traceable knowledge? We have this 5-step process depicted here. So first, we upload the SMPC PDF to the system. In the second step, we do AI extraction. A large language model identifies and extracts these key fields. And in the third step, we do semantic enrichment, where these fields are mapped to ontologies and integrated into a knowledge graph, which is shown here. At the top, for example, we have this SMPC document from which we extract the key field, which is the medicinal product here at the top left. And at the bottom right, we have these additional key fields like the disease indication, the vaccine classification, the active substance, the strength, the dosage form, shelf life, storage conditions, and so on. And some of these key fields we also map to ontologies, so they are classified either by the vaccine ontology, VO, on the top, or here on the right with this pink node by the disease ontology, Mondo. Or in case of antibodies or drugs, we classify the medicinal product directly by the NCI Thesaurus ontology shown by the red node, or in the case of small molecules, we classify by this chemical ontology, the chemical entities of biological interest. And then in this, after having built this semantic layer, we then build this web-based interface for accessing and interacting with the data directly. And in the last step, we have the traceability, where every data point links back to the original source. And now I would like to switch to the live presentation. Thank you. Yeah, for this demo, we have uploaded a set of 20 SMPCs from EMA, the European Medicines Agency, comprising small molecules, antibodies, and vaccines from different pharmaceutical companies. And what we do in the first step is we upload the documents. As an example, we can take here, we can drag and drop the file here, upload it, and process this SMPC. It's done. This triggers for LLM extraction of relevant fields and subsequent semantic enrichment. Now let's go to the medicinal products, which we have extracted here automatically from these SMPCs. So here we have, in the middle panel, we have the 20 medicinal products ranging from vaccines like Vaxchora for the prevention of cholera, or for the protection of human papillomavirus, Gardasil. But we also have drugs like Herceptin, therapeutic antibodies against breast cancer or colon carcinoma. And all of these medicinal products have here listed the key fields which we have extracted by LLMs, like the strength, the disease indication, cholera, the route of administration, oral, the marketing authorization, or the first publication year of the SMPC. On the right, we have then the full set of attributes like the dosage form, the container closure system, including also shelf life, 36 months, storage conditions, and so on. On the left side, on the other hand, we have the different filtering options for these medicinal products. So we can filter by vaccine or antibody classification, disease indication, or the dosage form, or the SMPC publication year. And now let's go for vaccine classification. For instance, we can either filter by viral vaccine or bacterial vaccine. If we go for viral vaccine, we reduce our number of medicinal products to 5. We have here Gardasil 9, Cervarix, Comirnaty for the protection of COVID-19, or here mRESVIA for RSV, respiratory syncytial virus infectious disease. Or we filter by vaccine classification of bacterial vaccine, and we obtain the vaccines for the protection for cholera, Neisseria, or here Streptococcus pneumoniae. We can also filter by antibody classification, and here let's go for anti-neoplastic antibody, and we get 2 therapeutic antibodies from Roche, here Herceptin for the treatment of HER2 positive breast carcinoma, or here Avastin for colon cancer. Or we can, in addition to filter by drug classification, we can also filter by disease indication. And here we can, for instance, go for infectious diseases or viral infectious disease. We get all the medicinal products for the viral infectious disease like COVID-19, RSV, and also influenza. Or we can go again for cancer. We go for disease classification, here cancer or neoplastic diseases. In addition to the 2 therapeutic antibody we just have seen, Herceptin and Avastin, we also have imatinib and Lumakras. Imatinib for the treatment of chronic myelogenous leukemia, and Lumakras for the treatment of non-small cell lung cancer carcinoma. And now let's see, we want to find medicinal products which have been published in the last 5 years, so from 2019 till 2024. We reduced the number of medicinal products to 11, and we are interested in administration or dosage form, dispersion for injection. And we go for vaccine classification, respiratory syncytial virus, and we obtain here mRESVIA from Moderna. We can go to the detailed report of this medicinal product. Here we have the full set of key fields which we have extracted, the strength 50 micrograms per 0.5 ml dose, dispersion for injection, route of administration is intramuscular, shelf life is 30 months, unopened, 6 hours at room temperature after thaw, and so on. And here in the middle we have the mapping to the ontologies. Here we have the mapping to the disease ontology entry of Mondo. Here we have all the synonyms associated with it, so we can do also synonyms in different languages. Here we have the synonym in Japanese. We have a short description of this disease. So infection with respiratory syncytial virus is based on an RNA virus of the genus pneumovirus, and it causes minor respiratory infection with rhinitis and cough in adults, but is capable of causing severe bronchitis and bronchopneumonia in young children. We also have a mapping to the vaccine ontology, where we have information of that this vaccine is licensed in the US. It's for respiratory syncytial virus. It's an FDA licensed vaccine, and so on. We also have here the link to the original SMPC, so the document, the PDF from which we have extracted here. In the middle, all these different key fields. We have here also an abstract. This is a Gen AI generated summary of this SMPC document, which typically is 50 to 120 pages. We have here a summary, the key messages, the clinical efficacy and safety, contraindications and precautions. Here we have the original document, which we can see in full screen. So here we have the summary of product characteristics, name of the medicinal product, MRSVIA, qualitative and quantitative compositions, posology, and so on. So that's the PDF from which we extract automatically all the key fields. And here on the right, we have a set of similar documents to the MRSVIA SMPC we are seeing here. So here we have another SMPC, also from Moderna, here Spikebox for the protection of COVID-19. But we also have scientific literature like a PubMed Central document or Medline abstracts or another EMA document, an assessment report for RSV vaccine, and so on. If we go back to the list of medicinal products, let's have a look to a small molecule like imatinib, which is used for the treatment of chronic myelogenous leukemia. As you see here, we have also a mapping to small molecule here, and we have all the links to the chemical information based on the interconnected knowledge graph. Here we have the structure of the imatinib, the drug, the active substance. Here we have all the synonyms. Here we have some information on the drug, so the approval in 2001. Then the chemical descriptors, INCHI keys, SMILES, cross-references to other databases like Campbell and KB and so on. Here we have the calculated chemical properties of this drug. And here we have similar structures, so we automatically run a similarity search. And here we also have the adverse events of this drug, like gastrointestinal stroma tumor, malignant neoplasm progression, and so on. In addition to search structurally through this filtering process, we can also ask natural language question by entering here a question into the search box. What does Mounjaro 2.5 mg dose quick pen solution for injection in pre-filled pen contain? This redirects us here to a document-centric page where we can get the answers. But here we have then the direct answer, Mounjaro 2.5 mg dose solution for injection in pre-filled pen contains 2.5 mg of tirzepatide in 0.5 milliliter solution. And this is extracted, as you see here, from this Mounjaro SMPC document. That's the evidence behind this answer. And here the system knows also what this is. This is a peptide, which you can see here. 3-tetrapeptide is a polypeptide. It's a hypoglycemic agent. It's also a anti-obesity agent. So based on the knowledge graph, we get additional information about this this drug. And yeah, why does this matter? So from documents to knowledge, so transforming SMPC into structured linked data builds this trusted foundation for regulatory and scientific intelligence. And with this semantic backbone, we link data semantically, ensuring traceability, consistency, and future-proof data management. And with that, I hand over to Eli, who will guide you through the next use case. Thank you.
Thank you, Angela. Now I will share my screen. Our second use case is focused around the fair metadata use and insights from clinical trials, demonstrating how a semantic layer can enable harmonized, CITES-compliant clinical data workflows. Each stage of clinical development is associated with its own set of bottlenecks. During the design phase, information often sits in silos, metadata is in reuse, which can slow things down the process. In the later stages, preparing data for analysis on machine learning can be time-consuming and complex, making it very difficult to generate insights efficiently. Ensuring effective trial comparison and data searchability remains difficult, largely because of inconsistent versions, distributed repositories, and restricted access rights. Having a semantic layer can significantly improve this process, as it enables the development of company-wide standards across studies, across therapeutic areas. It allows for an automated setup, predefines master data, which in turn makes downstream harmonization and analysis quite straightforward. Let's take a closer look at the following user story. As a data manager, I would like data from multiple silos to be automatically ingested and interconnected. You have maybe one database managing your SDTM data files, while sample and biomarker information sits in separate Excel sheets. The study protocol itself is in a separate location, store it as a PDF, and yet another database features your project, your contacts, and details about the lead scientists. Our system addresses this fragmentation by automatically ingesting data from different file types and sources, including external databases or data lakes, and bringing them all together into a unified knowledge graph. The knowledge graph itself, it interconnects the different entities and represents the relationship in between. For example, if you have a patient, in case of a clinical trial, you have a patient which participates in a study, which in turn is associated with a certain number of visits that are associated with certain time points. The study has a certain disease indication, and the patient itself is a participant in a certain trial arm that is associated with a drug that is in development and is tested as a part of a project. Experimental data such as vital signs and biomarkers or samples can be also included in your data sources, and the data that we have in our knowledge graph is also enriched by including public data sources such as clinicaltrials.gov or, for example, NCBI taxonomy. We can then navigate to our knowledge graph. And by navigating through it, we can see, for example, which drug is associated with trial arm, and with how many patients are associated in said trial arms, which in terms represents a linkage to our study and to the biomarkers. So we have, through this knowledge graph, a direct link from our drug to our biomarker. And we can see, for example, a list of biomarkers which were tested over the development of this drug. Now to go to a more of a direct example, using our approach, we can address the following use case. As a research scientist, I want to find all studies for hepatocellular carcinoma and sort them by the number of patients who received a treatment rather than placebo. I can then use those studies for further analysis. So for this, in this case, I would start by filtering for the condition, and then I can sort my number of results by the number of patients who received our treatment, and then we can extend our search, for example, by data, by additional criteria such as the availability of urine samples for my trials. In the end, I will get a result which features the trials which fit my choice of criteria. We can then use our knowledge graph to compare different trials based on certain criteria of interest and generate such comparison views. Or we can also find trials with similar content or similar metadata as the trials which is of my core interest. The knowledge graph combines and interconnects data from different sources. It maintains, however, strict control over data visibility through role-based access permission. Patient data is accessible only to users who are explicitly permitted to access these information. Other users can only see a more aggregated view of this information, such as the one that you can see here now. We can also perform advanced analytics and generate interactive heat maps to visualize complex experimental data, such as RNX pair pressure profiles. In the end, our knowledge graph is a central metadata hub that can be used as a single source of truth. We can use our data for downstream analysis and make your data AI ready. We can perform advanced analytics and offer also data to reporting via the other model integration. The benefits of data contextualizations are improved interoperability, seamless collaboration, and data and analysis integrity. We facilitate actionable insights by ensuring seamless data interoperability, which in turn enables teams to work together more efficiently and to have a shared view of data across the organization. In the end, this allows for better data management practices, improved data and analysis integrity. This transformative approach to data management helps foster a data-driven culture across the organization. With that, I would like to thank you for your attention